Hi, everyone. I'm Amy Goldstein, and it's my pleasure to join you for the special Mito 101 session for the Cure ARS Family Day. This will be a session that will last about a half an hour, and then after this session, you're going to be then joined by various people that have recorded sessions on each individual gene. So my session is about understanding mitochondrial disease and MT-ARS genes, basics for all. And these were my objectives to join for an engaging session exploring the fundamentals of mitochondrial disease and the biology of MT-ARS genes, learn how these genes are crucial to mitochondrial function, how they affect health, and the latest insights into this rare condition. This session is designed to provide a clear and accessible foundation for patients, families, and advocates. So I'm going to do my best to meet. So first, a definition. So when we talk about MT-ARS genes, these are within a larger family of disorders known as primary mitochondrial disease. So what do we mean by that? The definition of primary mitochondrial disease is that these diseases are caused by mutations that lead primarily or secondarily to oxidative phosphorylation dysfunction. I'll go over that in a moment, or other disturbances of mitochondrial structure and function, including perturbed mitochondrial ultrastructure, so how the mitochondria look, aberrant synthesis of cofactors and vitamins, or other impaired metabolic processes within the mitochondrion. So the mitochondrion are little organelles within the cell, and their job primarily is to make ATP, or our energy for the cell to run. And there's an outer membrane here, and then there's an inner membrane that form these little cristae here. And on the inner membrane, we have this electron transport chain made up of subunits 1, 2, 3, and 4. And then we have this other special subunit, subunit 5, and this is where ADP gets converted to ATP. This is where the energy gets made. And so this process of taking phosphorus and making ATP, we use oxygen, the oxygen we breathe, and the food that we eat to do this. This is the process known as oxidative phosphorylation, making ATP, using the electron transport chain to do that. And it does that by, you'll see all these other things getting imported. We have the Krebs cycle over here. This is making things that get imported over and feeds some of the subunits. We see coenzyme Q10 here feeding some of these subunits. Now there are, again, many arrows here and other processes that you'll see around the mitochondrion. Again, I wanted to point your attention here to the electron transport chain and oxidative phosphorylation, that whole process. There are other things that are happening here, reactive oxygen species, so extra oxygen or oxygen byproducts get made, and that can be damaging to the cell. Something that can help that is glutathione. So we like that to combat it. There are also other things that are happening. Free fatty acids are coming in using carnitine shuttles. And as I already mentioned, the tricyclic acid cycle or the Krebs cycles over here. There's also the urea cycle happening inside the mitochondria. Other things are getting imported through other chaperonins or transporters in the mitochondrial membrane. And then there are other things along this membrane that help stabilize it or help the mitochondria undergo fission and fusion, and that helps the mitochondria maintain its shape. And then we have the mitochondrial DNA, and that's undergoing replication and protein translation. And that's where some of the genes that we're interested in for the purpose of this talk are going to come into play later. But anything that affects any of these processes can lead to a primary mitochondrial disease. All right, so, and I didn't even talk about all of them. I'm going to show you another slide that talks about all the genes. Okay, what about the clinical features? So primary mitochondrial disease, I'm a child neurologist by training, and so I divide up the symptoms into those that are neurological and those that are non-neurological. A cardiologist might divide them into cardiac and non-cardiac, but I'm neurology, so here we go. So it turns out that the brain needs a lot of energy to run. And so if you had a primary mitochondrial disease and you weren't making enough energy, the brain is one of the first places that we're going to look for these symptoms. So you can have headaches, seizures, ataxia, migraines, dementia or cognitive changes, Parkinsonism, developmental delay, developmental regression, psychiatric issues or mood disorder. And so all of those things are possible. We can also have sensorineural hearing loss, and then we can have problems with the eye. So ptosis and external ophthalmoplegia, where the eye muscles aren't moving from side to side or up and down. And then we can have optic atrophy, where the optic nerve gets pale and small. Or we can have retinitis pigmentosa, which is a pigment that can turn up on the back of the eye and cause trouble seeing. Then we can have trouble in the spinal cord lesions. We can have trouble in the peripheral nerve and in the muscle. So in the nerve, we can get peripheral neuropathy, and that can lead to tingling and numbness or pain. And then in the muscle, we can have weakness, myopathy, and exercise intolerance. Okay, then we can switch over and talk about what's on the left side of your screen, the non-neurologic issues. So this can include respiratory issues, such as respiratory failure from chest wall weakness. We can have heart involvement, cardiomyopathy, or an arrhythmia or a conduction defect. We can have liver issues, such as elevated liver enzymes or liver failure. We can have kidney issues, including renal tubular acidosis, where the kidney will be spilling things such as bicarbonate or electrolytes into the urine. We can have kidney issues such as focal segmental glomerulosclerosis or renal failure. We can have adrenal insufficiency where you might need to take steroids when the body is stressed. We can have pancreas issues such as pancreatitis or diabetes mellitus. We can have major GI issues, anything from feeding issues, trouble swallowing, trouble with the stomach emptying, such as gastroparesis, poor motility. We can have something called intestinal pseudoobstruction, where the gut is acting like it can't move and it looks like there's an obstruction, but there's actually nothing truly blocking it. And then we can have failure to thrive, where it's difficult to get to normal body weight and grow properly. There can be problems with the sex organs, so we can have premature ovarian failure or lack of ovaries. We can have male infertility. And then there can be problems with the bones. We can have skeletal dysplasia, short stature, scoliosis, and in some cases, bone marrow failure. And I just went over some of the clinical presentations. This is another way to depict that. And this is a really famous slide that a lot of people use in their lectures, and it's from 1995. So we're going back a really long time. But the other thing that I like this slide for is it shows that a lot of the genes come from the nuclear DNA, our regular chromosomes, and they have to be imported into the mitochondria. And then we have the mitochondrial DNA, which donates some of its proteins that it makes. And that's what makes up these subunits that, again, reside on the inner mitochondrial membrane in the electron transport chain to undergo that oxidative phosphorylation to make ATP. And it's that ATP, that energy, that has to go and serve all of these organs for them to function properly. Okay, I told you I was going to show you a slide of genes, and there are at least 400 genes, if not more, already implicated in causing human primary mitochondrial disease. And the other thing I like about this slide is it really gets to the classification of mitochondrial disease. So we have the genes that are primarily responsible for oxidative phosphorylation, and we can see complex 1 subunits, and some of these are mitochondrial DNA, and some of them are nuclear DNA. And then the assembly factors for complex 1. We have complex 2, subunits, and assembly factors. Coenzyme Q10 genes, cytochrome C genes, complex 3 genes, complex 4 genes, and complex 5 genes. And again, all of the oxidative phosphorylation or electron transport genes, they have the complex subunits and assembly factors, and all of them are a combination of mitochondrial encoded genes as well as nuclear genes, except for complex 2, which is all nuclear encoded. The next category is mitochondrial dynamics, homeostasis, and quality control. And here we have genes that control things like the morphology, like the fission and fusion. Then we have genes that control mitochondrial DNA maintenance and gene expression. And again, some of these are mitochondrial encoded, all of the mitochondrial tRNAs, but many of them are nuclear. And again, the mitochondrial DNA is inside the mitochondria, but for it to be able to replicate and undergo protein transcription and translation, it needs nuclear genes to help that. And that's where these mitochondrial tRNA synthetases come in. So this is our family of genes that we're going to be talking about today. Some of the other categories are metabolism of cofactors, and those are here, metabolism of toxic compounds, and metabolism of substrates. And this is where, again, the TCA cycle that I pointed out earlier is, and some of those other transporters. Okay, so we have lots of families of genes, and again, this just shows some very nice packaging and classification of how we think about the mitochondria and how we think about the different genes and what their functions are within the mitochondria. And again, these are all primary. Now, this I took right off of the website for Cure ARS, and so this talks about what are the mitochondrial ARS genes, and you can read about this a little bit more on the website, but it talks about how rare mitochondrial disease is, but it actually isn't that rare. It affects 1 in 4,000 people, and this is probably one of the most common of the quote-unquote rare diseases that there is. It's more common than any other inborn error of metabolism. And I already talked about some of the symptoms, and this goes over some of the common symptoms that we see in mitochondrial ARS disorders. And what's really interesting about these disorders is that every gene is a little bit unique in that they don't all look the same. So if you have AARS2, you might look very different than someone who has IARS2. Now, there may be some overlap, right? If you're a child, a lot of children with these disorders have developmental delay, or they might have low muscle tone, but that may be where the similarities stop, because not everybody has, for example, white matter disease on the brain. Many people have a normal MRI, or some people might have what's called Lee syndrome, or some people with these disorders might have some other structural abnormalities in the brain. So again, there's a lot of differences here, and you're going to hear one by one each individual gene in the next series. Okay, so then there can be a pattern of organ involvement. Okay, so we can see encephalopathy, which just means a problem with the brain. Pathy means disease, encephalo means brain. So that can mean anything from stroke, seizures, or regression. I'm going to talk about this combination of deafness and ovarian failure, which is called quote-unquote Perot syndrome. I'll talk about that again. Cardiomyopathy, we'll have eye involvement, we can have lung involvement, and this combination of lung and kidney involvement together. We can have liver disease, we can have anemia and myopathy. And again, you'll see these acronyms. Sometimes we see these acronyms that are named for some of these disorders, and we can see skeletal dysplasia and neuropathy. And again, as I click along here, you're going to see some more of the genes appear, because we see that FARS2, RARS2, CARS2. We see that very heavily involved in brain. And again, some of the other genes just popped up because we can also see brain involvement. But you're going to see only 2 genes have been linked to Perot syndrome, LARS2 and HARS2. Neuropathy, we see GARS and CARS. So there isn't a number 2 for these. These have kind of dual function. All right, and more genes are going to come in as I keep clicking, because there are 3 genes in this family that can lead to cardiomyopathy so far: AARS2, YARS2, and VARS2. Now, if you have this condition, it doesn't mean you're going to get a cardiomyopathy. It means you might get a cardiomyopathy. And one of the things that we're working through is that dependent on where the actual mutations are within the gene, and we call that a genotype-phenotype correlation, or does it depend on something else, right? Maybe there needs to be a triggering factor that will bring that particular symptom out. A lot to learn. All right, there's a wonderful paper that I'll alert you to down here. It was published in Molecular Genetics and Metabolism, and it's called Mitochondrial Aminoacyl tRNA Synthetases in Human Disease. And I've taken some language from that paper, and I'll just read it out loud because I thought this was just a really beautiful description. So MTARSs are essential in the process of transferring genetic information from mitochondrial DNA to the complexes of the oxidative phosphorylation system. These synthetases synthetases perform an integral step in the initiation of mitochondrial protein synthesis by charging tRNAs with their cognate amino acids. What does that mean? I'm going to show you, and I found lots of fun pictures to show you. And I want to also say that so far, what we know is that all of these disorders, these primary mitochondrial disorders, these are autosomal recessive disorders, meaning you must have 2 copies of a pathogenic variant, one from your mother and one from your father, in order to have disease. That is different than what we call the cytoplasmic aminoacyltransferases, so the synthetases, the ones that do this in the cytoplasm, the protein synthesis that takes place for the other genes. So when they don't end in a number 2, these are autosomal dominant mutations, and these can cause neuropathy. So this is when, again, it doesn't end in a 2. AARS, YARS, and CARS, okay, and bifunctional GARS. There's not a separate one that does the cytoplasm and the mitochondria. The same synthetase does both. Okay, so let's start with some of these cartoons. So we have the complex. We have the aminoacyl tRNA synthetase, and this is an enzyme. Anything that ends in ASE is an enzyme. And we have a little tRNA, and this is a piece of genetic material. And what we want to have happen at the end is we want this tRNA to be attached to the amino acid that it's supposed to go with. And eventually this is going to get transferred to a ribosome, and this amino acid is a building block of protein, and we want to build a protein chain. Okay, but how are we going to get these two attached? This has to then go on its merry way. Well, that's what the aminoacyl tRNA synthetase is going to do, and it needs to do that with ATP. This is an energetic reaction that has to take place. So again, this is almost like a little key that has to bring these two things together. All right, so here's an example. And by the way, each one is specific for the amino acid that it's going to link with. Okay, so this tRNA is specific for methionine, and here's methionine, but this aminoacyl tRNA synthetase is also specific for methionine. Each one has to fit in just so. Again, this is a little bit of a different depiction. I really like it when they're like this, these little loops in this way. Okay, but anyway, here we have the one for methionine, our aminoacyl tRNA synthetase, and we have the amino acid coming in, we have ATP. Remember, we need energy for this to actually occur, and the tRNA comes in, and after the energy is utilized, boom, we have the tRNA connected to the methionine, and this can now go to the ribosome, and the methionine can get transferred to the protein chain that's being built, and the methionine will be in that right position. I'm going to show you more, okay, because each one of these gave me a little bit of a different depiction and understanding of what's happening. Okay, we have another ARS, okay, this is another one of our aminoacyl synthetases. We have an amino acid that's going to fit in, and we have the ATP that's going to fit in, and we're going to have the tRNA come along to pick up our amino acid, right? So again, we need the ATP to charge our amino acid, and we need the tRNA to come over and pick up our amino acid again so it can go on its way. I also found a beautiful YouTube video for anybody out there that likes watching YouTube videos. This was really well done. I forget now where this one was from, but the guy's voice was also very good. I will tell you, this was really well done. So this was a lecture I found on aminoacyl tRNA synthetase, and again, it describes how you have this time it's tryptophan, here's a tRNA, and this time it's showing you why are these so specific, because it's giving us the code, the 3-letter code for that amino acid, as it's done for in RNA and DNA. Every 3 letters is assigned to a specific amino acid, and this green, I want to call it a blob, but the green is representing the specific aminoacyl tRNA synthetase for tryptophan, and the tryptophan fits in, and the tRNA fits in, and again, these are going to get attached after ATP is added here, and boom, we have that high energy bond that ATP brought into our situation, allowing tryptophan to transfer and get attached to its specific tRNA with the help of the specific tRNA synthetase for tryptophan. And now this can go to the ribosome and tryptophan can get added to the growing peptide because it's the next amino acid in the chain. And finally, one more. Again, we have the tRNA, which is coded for by the mitochondrial DNA. We have the nuclear DNA that has made our aminoacyl tRNA synthetase, and we have the tRNA coming together. We have the amino acid which will join. We have the ATP, and then we have the amino acid joining the tRNA, and it's going to go and join the ribosome, and it's going to, here we have amino acids, and they are joining the growing peptide chain. These are each individual amino acids, and it's just going down the line. You can see all the letters here. For every 3 letters is coding for a specific amino acid, and these complexes are coming and sitting right here, and we have the amino acid joining the growing peptide chain. So this is protein synthesis, as told to you by a child. Okay. Here we have a list of all the genes, and again, I'm not going to go through them because you are going to be treated by many talks next, 17 of them, because we have 17 ARSs, and all of them will be for you. This is on the website as well, and this may be too small for you to read, but what I will tell you is there's a few things that I want you to know. Some of these are called COX-PD followed by a number, and this stands for combined oxidative phosphorylation. Okay, combined oxidative phosphorylation and the number. And that is the name that was given to the disease, sometimes even before we knew what the gene was responsible for that condition. And when we say combined oxidative phosphorylation, sometimes what that means is that we may have done historically a muscle biopsy and looked at that electron transport chain and said, there's not enough complex 1 and there's not enough complex 3. We have a complex 1 and 3 deficiency. It's a combined oxidative phosphorylation defect. And we already have discovered 1, 2, 3, 4 of them. Let's call this one number 5. And there you go. And that's how they named conditions before we knew what the genes were for those conditions. So don't let that nomenclature throw you. It's going to be in here in a couple places. Perot syndrome, I'm going to talk to you about on the next slide. There's also a few of these that have been called Charcot-Marie-Tooth, and I think that has been more for the autosomal dominant forms. Charcot-Marie-Tooth is also a historic designation, but we still use it, and there are many forms now of Charcot-Marie-Tooth, some of which we know the genetics of, but these are, I think, 3 French physicians, that Dr. Charcot, Dr. Marie, Dr. Tooth, that noticed a peripheral neuropathy, and the arches got very high, what we call pes cavus and hammer toes, and loss of reflexes. And if we see that, we say, oh, this looks like CMT or Charcot-Marie-Tooth, but just realize there are now many different forms. They now have numbers and letters, so somebody might have CMT type 1A or CMT type 3B, but now we know the genes for these, okay? So sometimes things are still called by their old nomenclature. Another thing that you might see is this DEE, a developmental epileptic encephalopathy. What does this mean? So DEE, the epileptic encephalopathy means that a child or adult might have so many seizures and the EEG may be so active that it's hard to have normal activity in between and therefore hard to undergo normal development. And if you can help that child stop having seizures with medication and clean up the EEG a little bit, they actually might start making better progress. And there are many genes that cause this, not only mitochondrial genes, but other epilepsy genes. So this is another whole family of disorders. And then there's another abbreviation buried in here, PCH, and that stands for pontocerebellar hypoplasia. which is where part of the brain doesn't grow properly. Hypoplasia means that it's just small from the beginning. And I told you, sometimes we see differences in the brain. Sometimes it's white matter, changes. Sometimes it's Lee syndrome, which are lesions in the basal ganglion brainstem. But sometimes it's structural brain malformations, and PCH is one. Okay, Perot syndrome, I told you there are two causes of that in this family of MTARS, LARS2 and HARS2. But look, there's other non-mitochondrial causes as well. So there are 6 forms of Perot syndrome. So what is Perot syndrome? Perot syndrome is, according to OMIM, the Online Mendelian Inheritance in Man website, a sex-influenced disorder characterized by sensorineural deafness in both males and females and ovarian dysgenesis in females. This was first described by Perot in 1951 in 2 sisters who had deafness, and they lacked ovaries, and their parents were cousins. Now, some people with these gene mutations have neurologic manifestations, including mild intellectual disability and cerebellar and peripheral nervous system involvement. But this was noted by Pierce et al. This is a paper that was published in 2010. But Pierce noted that Perot syndrome was very heterogeneous. So he decided that we need to classify Perot syndrome into type 1, which was static, non-changing, without neurologic disease, and type 2, which is progressive neurologic disease. And I will also say that just because, let's say that you have a child that's been diagnosed with Perot syndrome, it does not mean that they will have all of the manifestations of what you read about. All of these disorders are heterogeneous and variable. Okay, what about treatment options? So I'm at CHOP, the Children's Hospital Philadelphia. We use a mitochondrial medicine regimen, and we personalize that based on certain lab tests and what the genotype of our patient is and what their phenotype is. What are their symptoms? So as you can see, when people have we use a core medicine regimen, so a multivitamin, a B complex, coenzyme Q10, and some antioxidants for that reactive oxygen species that I showed you, the ROS, in one of the earlier slides. But if somebody has complex 1 deficiency, we use some niacin and riboflavin. If somebody has lactic acidosis, we use biotin and thiamine. If somebody has mitochondrial depletion or Kearns-Sayre white matter disease, we add folinic acid. So you can kind of see how we personalize it. But I also wanted to touch on amino acid supplementation. This is one of the proposed treatments for MTARS disorders. So I want to talk about that. And we heard yesterday that there's a protocol that's been developed by Rita Horvath and her colleagues in Cambridge, and they are planning to start a study in April of 2025. So what does that mean? We know that MTARS disorders, the job of the synthetase is to link the amino acid with its cognate tRNA, right? I want my alanine connected to my tRNA for alanine to use the amino acid. So people have published this, and there's been a lot of excitement about this, right? What if you have FARS2? So the other thing is that these letters stand for the amino acid, and because there's more than one P, you have to use different letters. So phenylalanine is what the F stands for. Well, if we give somebody phenylalanine, is there a concern? There's actually a disorder where people have too much phenylalanine in their system, and it's called PKU. And that causes intellectual disability. So it's not a great thing to have too much phenylalanine. So number one, if we're going to do this, we need to know what are the absolute correct doses to use. And number two, we'd like to do it using a protocol so that we know what we're following. How are we going to know if people get better? We don't want people just grabbing amino acids off the shelf and doing this on their own. But this was a paper that was published I think in 2023, and it showed that by giving oral phenylalanine, they were able to treat an individual with FARS2, and they improved in all areas tested. gross motor skills, movement, postural stability. And then when they were without supplementation, she lost newly acquired skills and then regained them upon restarting supplementation. So this was really nice evidence. And again, this has been a proposed therapy. Can we give the cognate amino acid to people with mutations in an aminoacyl tRNA synthetase disorder? So this is my analogy for how this would work. We all know the claw machine, right? And so if the claw machine is your aminoacyl tRNA synthetase and the ATP that's needed is the coin you put in and the electricity to make this whole reaction happen, and the claw is your tRNA, and the little prize is your amino acid that you want to get attached here. Well, what if the claws, what if the machine's broken? How are you going to get the prize in that claw? Well, what if we just fill up the machine with more prizes, more amino acids? Aren't you eventually going to have something end up in that claw, even if the machine is broken, right? So that's the premise behind giving amino acids, at least my simplified version. However, the question is, does it matter where the variants are? Are there some variants? Okay, this is the FARS2 gene. I'm just using this as one example. The mutation can be anywhere in this gene. And sometimes it could matter if the variant is in one position or the other, as far as how receptive is it going to be to amino acid therapy. All right, so there's a lot of I'll end with, there are a lot of different symptoms for mitochondrial disease, as I've mentioned, and it takes a village. You may need many different specialists to handle those symptoms. There's also a lot of support out there. So you have the Cure ARS Foundation. There's also the UMDF and Mito Action. There is the Mitochondrial Medicine Society, and there are clinics in the US and in other countries. And we hope that you have mitochondrial disease doctors who understand your. And with that, I want to thank you for your attention. Please power through the next talks because we want to help these sick mitochondria become healthy, strong mitochondria. And thank you again to the Cure ARS Foundation, and thank you for your attention today.